And we move on to the next topic of discussion. And thank you, Dr. Nirupama, for talking about the Wnt, the notch, and also the fate to commitment. And this is what we will be discussing in the hepatoblastoma. I do have to admit that this is also a new topic to me, unlike the HCC. So what I have divided the slide, the outline of the talk is, the first is a prelude, why this topic was chosen for the discussion. And the points which I want to cover is, from where are these hepatocytes, hepatoblastomas originating? Why is the Wnt and the beta-catenin the holy grail? And why we need to now move on to the epigenome. So in the prelude, this is a slide borrowed from Dr. Archana, and if you see, this is the data from ILBS. Almost 97% of the tumors are hepatocellular carcinoma, and this includes both the adults as well as the pediatric group. And when you turn to look at the hepatoblastomas, they're only 3%. Maybe this can go up to 5% if you include all the cases. But what this tells us that indeed this is rare. If this is rare, then what is unique about it? So the first thing for my understanding was, how do I differentiate a hepatoblastoma from a hepatocellular carcinoma? So for this, I think you need to have three different insights: a pediatric view, a pathologist view, and a basic biologist. So if you compare the two, and this is the hepatocarcinoma, although mostly in the adults, but you will see that there are also in the pediatric group. The hepatoblastoma is unfortunately in the adorable group of 0 to 4 and also the 5 to 15 years. The HCC mostly in the older age group. And what is interesting and common between the two, but that's what I found it in the literature. And this is actually the data which is from the Japanese group, that they have also gone up to 16, and there are in fact very rare reports on hepatoblastomas even in the adults, which I'm not going to cover up. So one thing which is common between the two is this so-called congenital abnormalities, which the pediatricians know much better. And from an etiological perspective, low birth weight and parental smoking is an interesting fact. I did not come across alcohol. In fact, few of the literature says that alcohol doesn't have any association. That is, paternal drinking of the alcohol not associated with hepatoblastomas. Compare this with the hepatocellular carcinoma, which you as clinicians know much better. Now, when it comes to the pathology, and that's where much of the work comes from, that the hepatoblastomas usually are fetal and embryonal. That tells that there is something going haywire during the development of the liver itself. Unlike the hepatocellular carcinomas, which can be poorly or well differentiated, and they can also have a so-called stem cell features. But the most intriguing part is that when you talk about the genetics, it's actually at a very low tumor mutation burden of only 2 to 5 prominent drivers, unlike the hepatocellular carcinomas, where the tumor mutation burden is more. So from here now I'll take on towards what, okay, I didn't see that, but I'll come to some genetic abnormalities which will answer your question. So if I have to now cover all these parts up, so the questions are the cellular origin, which are the pathways dominant, and the pathways which are dominant are the Wnt and the notch. And these are arising from the progenitor. So this block here talks about the hallmarks. And then, of course, we need to move, since the mutations are so rare, but the pathologist will tell you that it's a complex disorder. So we need to now focus on the epigenome and the transcriptome. And who do we take the help from? I've taken the help from a Drosophila. So before I go to the Drosophila story, this is what talks about the intersection between the liver organogenesis and cancer. And this is very important because hepatoblastoma being a pediatric tumor, you need to learn about the liver development. Dr. Anupam is an expert here, not me, but what I've done is a very superficial slide in a very simple way talking about directly from the endoderm, the liver lineage starts. And you have a so-called multipotent progenitor, a bipotent hepatoblast, and finally, this is where the commitment takes place, hepatocytes and the biliary cells. But do you think that the commitment is only restricted here? No, in fact, there are in between many transitional states which we do not know of at all. Now, in case of the hepatoblastoma, what is happening is that in this commitment stage, when it is going towards either the hepatocytes or the biliary, there can be certain mutations taking place. And so that means now the fate has been sealed here. So if the fate is sealed here, you have an embryonal or a fetal type. However, if something goes wrong in the hepatocyte or the biliary lineages, that's where you have the hepatocellular carcinomas and the cholangiocarcinomas. So the question is, what is the genetic makeup during this commitment thing? And what I want you to all remember is that from the endoderm going up to the last stages, you have the so-called prominent highlighted mark, which are the Wnt and the notch. And I'll come to that in a minute. Now, this is just a very superficial again. Recently we started doing some sequencing in the HCC, and here there is one, so this is only one representative slide, an adult and a 14-year-old child, and when they were sequenced, we found that there are at least 10 to 15 mutations per tumor. Now compare this with what is there in the literature for the hepatoblastomas, only 2 to 4 mutations. And in HCC, it can go up to 30. So there are numbers of these passengers and so-called driver mutations. So what are the driver mutation in hepatoblastoma if it's only 2 to 4? The most prominent driver, as you can see, this is CTNNB. This is a biggest study from the Japanese group, and what they've done is they've taken so each of these columns up here represents one individual, and this represents up here, each of these rows is a gene, and you can see that almost 77% of the tumors have a beta-catenin mutation. What is interesting to know here is these are all somatic variants, very low frequency, highest frequency of beta-catenin. Now you also see that there is an underlying background which I did not mention about, is this germline variance. And hepatoblastoma is seen in something like familial polyposis. You as a clinician know it much better. In Leaf-Romanes syndrome also they may have, that family may have, and also to some extent those where there is a DNA damage checkpoint responses. But what is most intriguing is 78 to 95% in the literature reports to the CTNB. Now you see there are two colors here. One is a light green and one is a dark green. That means light green means that there is a missense mutation. That means very small mutation, A changing into a T. Unlike very large or bigger frame shift mutations also can occur. And this kinds of gives the dichotomy, which will come to the next slide. But before I go into that, this is from a pathologist's perspective, and as sir was telling you, where exactly is the commitment failing in case of hepatoblastoma? The commitment is failing someplace here when it is still in a very primitive multipotent state or when it is in a bipotent state. So depending on from where it is getting arrested, and this has been worked out very nicely by Dr. Saddarshan Monga using the mouse models, you have the small undifferentiated type. This is the human pathology, embryonal type, crowded fetal or fetal. So basically you can divide it into two groups, an embryonal group and a fetal group. Now this is for the histopathology. What about the basic sciences of it? Now this is again in a very simple way explaining these tumors have been divided very broadly into two types. Those where the CTNB mutation or the beta-catenin mutation is only missense, that is A changing to a T in the exon 3 region or in the exon 10 or 12. You can also have bigger lesions. Now the bigger lesions, if there are there, predominantly the notch pathway somehow takes over. So that means now there is an epigenetic wiring occurring. If there is a small missense mutation, the metabolic pattern is such that it is much more glycolytic because CTNNB or beta-catenin turns on the GLUT3, and GLUT3 is mostly of fetal origin, mostly expressed in the fetal, more of the glucose intake, cells are less of OXPHOS and more of glycolytic. So the pathway which is predominant in the embryonal histology is the Wnt. In the fetal, it is the notch. Now, so now this is all gene expression studies. So the gene expression studies are showing that there is less expression. It doesn't mean there is no expression. These are all relative expression. The genes for the glycolysis are less expressed. So the interpretation is that it is going more into an OXPHOS. This is in the fetal type, which is in the later stages. So that's what you would expect. In the embryonal type, these are actively dividing. Now, embryonal is also an analogy to what is happening in a cancer, where it's very fast, and maybe they are more glycolytic. But this is only from the data which is there. So I have just made it into, and the embryonal type are the more primitive, more aggressive, and the fetal type are the less aggressive. So now the same thing has been divided here. Now you can see, you can pick out your genes here. There's a 16 gene expression panel, and this is an old work which was published. And this is, so that means so now what is happening is beta-catenin is not there at all. So some pathway has to take up. So which is the pathway which is taking up? It's now taking the pathway towards the notch. Beta-catenin is more in, it's more in a crime with the Wnt here, at least in the hepatoblastomas. So now you see that where the molecular heterogeneity came at the level of the cell, as Dr. Nirupama was discussing in the previous slide, then at the type of the mutation and then the type of the signaling event. So that's what we need to remember with hepatoblastoma. Now there is this molecular heterogeneity from a basic perspective. How do you translate into the science? And this is what this long back paper said, that the embryonal one have a very bad prognosis compared to the fetal origin one. Now, what is this Wnt and notch all about? So first I'll talk about the Wnt. And so Wnt is a glycoprotein. Now this is actually secreted from the mesoderm, at least during the early development, and at least macrophages and the other immune cells or the endothelial in the later part of the development. The Wnt is a glycoprotein, it's a growth factor. It turns on a gene called the beta-catenin. This is what is actually mutated in the hepatoblastomas and also in the cancer, in the adult. Now this is how the HCC looks like, the beta-catenin looks like, supposedly present on the membrane, and when it is mutated, it actually goes inside and sits in the nucleus. And this pathway is a normal switch, very important during the liver development for the stem cell species fate, but something goes bad, that's where the cancer starts. So let's see what this term WNT means. I'm calling it as a Wnt. Actually, I should have called it a WNT. Why Wnt? This is actually coming from two things. So for that, we need to take help for the Drosophila. So everything that you see in liver development is actually happening in a Drosophila. That's what, there's a famous statement, nothing in biology makes sense except you need to look everything in terms of, in the light of the evolution. And both the Wnt and the notch are dominant in the stem cell compartment during the development, and it is the mesoderm that is giving out all these factors and then telling the species, the lineage towards whether it is going to the liver or to the pancreas or whatnot. Now, where is this term coming from? This term is coming from integration of two genes. W-wing integration from the mouse model. So this is a very interesting story buried in the history now. But what is missing and very sad about it, the wingless mutation was first discovered in India way back in 1973 in Pusa, but very less cited. But this wing, so this is a wingless from Dr. Sharma's group in Pusa, and you see that either it is rudimentary there or it is not present. This is a normal wild type fly. You see the fly here, the one with the wingless, this has a very red eye, so that means there is a developmental defect even in the eye of this Drosophila. Now this was happening in 70s and 80s, and at the same time what was seen in case of the mouse, and this was a totally independent work which was being done, they discovered a mouse virus. This mouse virus was integrating into the mouse genome, leading to mammary tumors. Now where was this mouse virus? And this was a work done by again a Nobel laureate, Harold Wormus. They found that this virus is actually going and integrating into the site, and that time it was called as the INT site. So they gave the name, the gene, the INT. Later on, when more of this genetics thing came up, they found that this Wnt is nothing, this INT is nothing, but it is actually the analogous gene present in the Drosophila. So they termed this as wingless and INT as the Wnt, and there are some 20 such genes which are very important from a stem cell to the development of an organism, and this got a Nobel Prize in Medicine in 1995. And there are some 1 to 19 Wnts actually in the human. So now back again to the story of the hepatoblastoma. So now in a normal development, Wnt which is there, it actually turns on the beta-catenin. Beta-catenin is present in the cytoplasm, and it gets degraded. When Wnt is on, this beta-catenin goes into the nucleus. Now when Wnt is in the nucleus, then it tells the cells you have to do a survival, you need to divide out, and you also have to do a commitment. And this is what is happening in a normal, and then the switch is turned off. But if the switch is turned on altogether, you really don't need the Wnt here. What you need to do is just turn the switch on by truncating mutation or by doing a simple mutation into the beta-catenin gene, and this pathway is all the time on. When this pathway is all the time on, there is a failure to commitment to either a hepatocyte or a cholangiocyte lineage, thereby the cells getting arrested into a tumorogenic phenotype. So constitutive activation of the Wnt signaling is what is up. So what is happening is if it's early in the embryonal, then it is the Wnt. Later in the fetal type, it is actually the notch. So now, okay, so one is you turn on the Wnt, and that's what the beauty of the nature is. You mutate beta-catenin, you end up getting the hepatoblastoma. You don't need Wnt. Now, neither do you need Wnt, neither do you need the beta-catenin mutation. What you need is just get rid of this APC. And APC is actually a gene which is adenomatous polyposis coli in the colon cancer. And this is a policeman that keeps in check the beta-catenin. The policeman is gone, no need for the growth factor. So what will beta-catenin do? It will go inside the nucleus. So this is another pathway where you can have a Wnt activation even in an absence of a beta-catenin. Now there's another pathway which I said was notch. Again, we take help from the Drosophila. Now you see this is a wing pattern of a wild type, and this simple notch here, not big difference. But this has made a big difference with respect to the development. How? This notch is actually falling into a very important pathway which is called as the notch ligand pathway. This is the notch. There is a receptor. This receptor binds to a ligand which is present in another cell. They both dimerize. Then once they dimerize, there's some enzymatic reaction takes place. The lower part is released out of the notch. It goes inside. Now the notch signaling from a pediatric point is important for the Alagille syndrome, but it has been also reported to be important in the hepatoblastomas. So now you can have either a Wnt activation or a notch activation. Now you may ask, how is this notch getting activated? Because in all the genetics, it's only the beta-catenin and the Wnt which are more prominent. So let's ask the question how this is happening. So just to summarize the first part, it's a genetically stable, unlike the HCC. Predominant is the beta-catenin arising from the stem cell, and that one gene itself is enough to do all these histological diversities. Now we move on to the notch. To understand notch, we need to little bit understand the epigenome before we go into that. Now this has been covered earlier by Dr. Vijay and all a lot on the epigenetics. This is a very simple way. So now you don't blame the gene mutation, now you blame the environment. So let's say you have a sequence, the same sequence is here, except that I have put some red flags. So these are some CG islands. When these are flagged with some red marks, which is the methyl, which can come either from a nutrition, from a diet, it can also come from something like a toxicant like a metal or a paint, it gets hypermelated. So that means it's the same sequence, no mutation, still the product or the gene is not made. The gene is not made because these are now methylated. So now if you turn it back, it can go back to its original. So unlike the genetic change, which is actually kind of fixed, the epigenetic change is actually very dynamic, and it can reverse out. Now, this part was covered earlier also by many people in the class, but what I want to cover with respect to hepatoblastoma is a very interesting thing which is called as the imprinting. But before we know the imprinting, let's talk about the normal biallelic expression which each one of us has. One parental or one gene present on the maternal chromosome and a paternal chromosome, and you are all diploids. But what happens that there's a set of some 80 genes which are actually imprinted, and these imprinted genes are very important for development. So what happens is that the father gets flagged for one of the genes. And this methylation comes, the father's allele is switched off, the mother's allele is on. This is very important from an evolutionary concept. I'll come back to that later. So that means what is happening for the imprinted genes is just because of a methylation, only one gene is working. So now you would ask that we all know about the diploid status is lost. But that's true, the diploid status is actually lost here. But what if something goes wrong, this methylation took place? Now, the gene which has to be present only in one copy is now getting expressed in two copies, and that's where that becomes an overdose. When this becomes an overdose, then that's where the system goes cranky, and there are many genetic abnormalities. Okay, so now I'll give you an example. Can you hold on to a thought? I'll just say. So now you have the so-called uniparental disomies. You're getting now both the genes from the mother. Now when you're getting both the genes for the mother, and the mother's gene is on, so what will happen? You have an overdose. Let's say the father's gene got switched on. Again, you have an overdose. So these genes are very important and crucial for a normal development. Why this is so? this is an interesting concept, that there is actually a tug of war between the genes from the maternal and the paternal side for the number of progeny, the type of progeny that you are going to have, and also the development. So there is actually a tug of war. These are all evolutionary concepts you may or may not believe. So you can have imprinting in both the ways. You can have a paternal imprinting, you can have I'm sorry about this is not material, this is maternal imprinting. And the gene which is the notch, that is paternally imprinted. That means the notch is only given by the mother. But if there's a uniparental disomy, that will what will happen is lot of notch will be done. Similarly, insulin growth factor and ACL2, I'll go into the second slide, and maybe from here you will understand it better. So now this is from the maternally imprinted genes. Now you look at the IGF locus. The male, so I should not call it like, so let's say the paternal gene is not flagged. The maternal gene is flagged. So what will happen? IGF, as you all know, is important for the fetal development. So IGF will be produced, there will be normal growth. Now let's see what happens if your the maternal gene for some reason is also turned green. So now you will have overdose of this insulin growth factor. Now if there's an overgrowth of insulin growth factor, what will happen? The growth will be humongous. And this is what has been seen in this thing, that if you go into the lower organism, something like a mice, which is having a brood of, let's say, 8 to 10 pups. Now the mother has to equally make sure that all the pups are alive, all the pups are growing well. At the same time, they're not taking away her nutrition also. But from the perspective of the paternal, it's actually the speciation which is more important. So what the nature did was, the mother very silently or cleverly turns off her gene. Now the same thing can happen the other way around with the paternally imprinted genes as well. And it's very interesting to know that there is this locus, the 11 chromosome locus, where there are two genes. One is this insulin growth factor, other is this transcription factor. Now you think both of them are in the same locus. Let's say that there was an environmental pollutant, or there was something to do with the diet. This locus got turned on. So now what will happen? There will be excess growth. Now if this can be now related with the liver development, this is what is happening, that there will be more proliferation of the stem cells. So one is a Prader-Willi syndrome, the other is the Beckwith-Weidman syndrome, and the other is the Russell syndrome. So Beckwith-Weidman is big. Russell syndrome, it is actually the paternal gene which is active, so the baby doesn't grow at all. So now so that was with maternally genes. Now this is the other gene. The notch is actually a paternally inherited gene. And why all this? I'm not going into the detail. People have done a lot of gene expression. It's important clinically. Now, based on the epigenome of this, people have classified as risk status 1, risk status 2, and risk status 3, looking at the epigenetic profile of this particular locus. And this makes sense because this is what you want actually in the clinic to actually do the prognostication. Okay. Now there is another new thing which has come up in the literature, and this is this gene called the NADPH quinone. I think I'm almost done now. So this NADPH quinone, this is a Japanese work. They are again classifying all. So now from one gene, now people are going into molecular stratification of the tumors. And here there is most of a ROS production because this gene is flagged. It becomes hypomethylated, so more expression. Now more oxidative damage. This is what they hypothesize that could then lead to a change in the developmental pattern. So now they are calling it as E1, E2 and an F cluster. E1 and E2 in the embryonal, and the cluster F is in the fetal. So this is the summary now, that from what I understand, you have only one gene. If this is the culprit, then either it goes into a Wnt or a notch, depending on what is the type of mutation. So we need to look at here. You can also do an expression profiling, and you can have a profiling for the fetal versus the embryonal, for those places where they're kind of a mixed. So you have an mRNA, and then you have an epigenome or the genome genetic locus for the imprinted loci. And at least these are 3 or 4 hotspots that one can do about. So where is the science taking us now? There are challenges. There are a number of clinical trials, and I'm not even going into that. All this talks about the beta-catenin pathway, and there are so many of these drugs. But will any of them work? We do not know, because this is such an important pathway that it is actually occurring in the stem cell. So now if you're targeting it out, what happens to the bone marrow stem cells? That also need Wnt. What is happening to the skin? What about the hair? So all this kind. Now with respect to the algyls, they are actually having an antibody that can actually work on the jagged and the notch. So maybe this can be also helpful. So this is again the summary that I've talked about the same. And where are we? So I think now, thanks to the genomics department in Albius, we can actually do a lot of NGS, and Dr. Pramod is also here. He's very good in doing all the bioinformatics, but that's an expensive thing. With the limited sort of money, we can now actually do the beta-catenin mutation, which we did together with Dr. Archana. This is from simple paraffin-embedded slides, 4-micron thin sections, but this is in HCC. So there are pros and cons. NGS, of course, is much better, and for pediatrician, you can take it out. If you have a fresh frozen, then you can look for the long deletions. So this is what we need actually if you can develop a pediatric cohort, because the number are very small, and what you need is actually a consortium for these imprinted genes. This is a very interesting area.